Cell Biology. From Wikipedia, the free encyclopedia. This article is about the term in biology. For other users, see cell disambiguation. Cell Wilson 1900 Figure 2.jpg Onion, allium, cells in different phases of the cell cycle, drawn by E.B. Wilson, 1900 Cell types. SVG a eukaryotic cell, left, and a prokaryotic cell, right, identifiers th. H1.00.01.0.00001 FMA 68646 Anatomical terminology, edited on Wikidata. Structure of an animal cell. The cell, from Latin cella, meaning small room, 1, is the basic structural, functional, and biological unit of all known living organisms. A cell is the smallest unit of life that can replicate independently, and cells are often called the building blocks of life. The study of cells is called cell biology. Cells consist of cytoplasm enclosed within a membrane, which contains many biomolecules such as proteins and nucleic acids. 2. Organisms can be classified as unicellular, consisting of a single cell, including bacteria, or multicellular, including plants and animals. 3. While the number of cells in plants and animals varies from species to species, humans contain more than 10 trillion, 1013, cells. 4. Most plant and animal cells are visible only under a microscope, with dimensions between 1 and 100 micrometers. 5. The cell was discovered by Robert Hooke in 1665, who named the biological units for their resemblance to cells inhabited by Christian monks in a monastery. 6. 7. Cell theory, first developed in 1839 by Matthias Jakob Schleiden and Theodor Schwann, states that all organisms are composed of one or more cells. That cells are the fundamental unit of structure and function in all living organisms, that all cells come from pre-existing cells, and that all cells contain the hereditary information necessary for regulating cell functions, and for transmitting information to the next generation of cells. 8. Cells emerged on Earth at least 3.5 billion years ago. 9, 10, 11. Contents hide. 1. Anatomy 1.1 Prokaryotic cells 1.2 Eukaryotic cells 2 Subcellular components 2.1 Membrane 2.2 Cytoskeleton 2.3 Genetic material 2.4 Organelles 2.4.1 Eukaryotic 2.4.2 Eukaryotic and Prokaryotic 3 Structures outside the cell membrane 3.1 Cell wall 3.2 Prokaryotic 3.2.1 Capsule 3.2.2 Flagella 3.2.3 Fimbria 4 Cellular processes 4.1 Growth and metabolism 4.2 Replication 4.3 Protein synthesis 4.4 Motility 5. Multicellularity 5.1 Cell specialization 5.2 Origin of multicellularity 6 Origins 6.1 Origin of the first cell 6.2 Origin of eukaryotic cells 7 History of research 8 See also 9 References 10 Bibliography 11 External links 11.1 Textbooks. Anatomy comparison of features of prokaryotic and eukaryotic cells Prokaryotes Eukaryotes Typical organisms Bacteria, archaeoprotists, fungi, plants, animals Typical size 1 to 5 meters, 12, 10 to 100 meters, 12, type of nucleus nucleoid region, no true nucleus with double membrane DNA circular, usually, linear molecules, chromosomes, with histone proteins RNA/protein synthesis coupled in the cytoplasm RNA synthesis in the nucleus protein synthesis in the cytoplasm ribosomes 50S and 30S 60S and 40S cytoplasmic structure very few structures highly structured by endomembranes and a cytoskeleton cell movement flagella made of flagellin flagella and cilia containing microtubules, lamellipodia and filopodia containing actin mitochondrion and 1 to several thousand chloroplasts none in algae and plants organization usually single cells, colonies, higher multicellular organisms with specialized cells cell division binary fission, simple division, mitosis, fission or budding, meiosis chromosome single chromosome more than one chromosome membrane and membrane bound organelles cells are of two types. Eukaryotic, which contain a nucleus, and prokaryotic, which do not. Prokaryotes are single-celled organisms, while eukaryotes can be either single-celled or multicellular. Prokaryotic cells. Main article. Prokaryote structure of a typical prokaryotic cell. Prokaryotic cells were the first form of life on Earth, characterized by having vital biological processes including cell signaling and being self-sustaining. They are simpler and smaller than eukaryotic cells, and lack membrane-bound organelles such as the nucleus. Prokaryotes include two of the domains of life, bacteria and archaea. The DNA of a prokaryotic cell consists of a single chromosome that is in direct contact with the cytoplasm. The nuclear region in the cytoplasm is called the nucleoid. Most prokaryotes are the smallest of all organisms, ranging from 0.5 to 2.0 meters in diameter. 13. A prokaryotic cell has three architectural regions. Enclosing the cell is the cell envelope, generally consisting of a plasma membrane covered by a cell wall which, for some bacteria, may be further covered by a third layer called a capsule. Though most prokaryotes have both a cell membrane and a cell wall, there are exceptions such as mycoplasma, bacteria, and thermoplasma, archaea, which only possess the cell membrane layer. The envelope gives rigidity to the cell and separates the interior of the cell from its environment, serving as a protective filter. The cell wall consists of peptidoglycan in bacteria and acts as an additional barrier against exterior forces. It also prevents the cell from expanding and bursting, cytolysis, from osmotic pressure due to a hypertonic environment. Some eukaryotic cells, plant cells and fungal cells, also have a cell wall. Inside the cell is the cytoplasmic region that contains the genome, DNA, ribosomes and various sorts of inclusions. 3. The genetic material is freely found in the cytoplasm. Prokaryotes can carry extra chromosomal DNA element called plasmids, which are usually circular. Linear bacterial plasmids have been identified in several species of spirochete bacteria, including members of the genus Borrelia, notably Borrelia burgdorferi, which causes Lyme disease. 14. Though not forming a nucleus, the DNA is condensed in a nucleoid. Plasmids encode additional genes, such as antibiotic resistance genes. On the outside, flagella and pili project from the cell's surface. These are structures, not present in all prokaryotes, made of proteins that facilitate movement and communication between cells. Eukaryotic cells. Main article. Eukaryote structure of a typical animal cell. Structure of a typical plant cell. Plants, animals, fungi, slime molds, protozoa, and algae are all eukaryotic. These cells are about 15 times wider than a typical prokaryote, and can be as much as 1,000 times greater in volume. The main distinguishing feature of eukaryotes as compared to prokaryotes is compartmentalization, the presence of membrane-bound organelles, compartments, in which specific metabolic activities take place. Most important among these is a cell nucleus. 3. An organelle that houses the cell's DNA. This nucleus gives the eukaryote its name, which means true kernel, nucleus. Other differences include, the plasma membrane resembles that of prokaryotes in function, with minor differences in the setup. Cell walls may or may not be present. The eukaryotic DNA is organized in one or more linear molecules, called chromosomes, which are associated with histone proteins. All chromosomal DNA is stored in the cell nucleus, separated from the cytoplasm by a membrane. 3. Some eukaryotic organelles such as mitochondria also contain some DNA. Many eukaryotic cells are ciliated with primary cilia. Primary cilia play important roles in chemosensation, mechanosensation, and thermosensation. Cilia may thus be viewed as a sensory cellular antennae that coordinates a large number of cellular signaling pathways, sometimes coupling the signaling to ciliary motility, or alternatively to cell division and differentiation. 15. Motile cells of eukaryotes can move using motile cilia or flagella. Motile cells are absent in conifers and flowering plants. 16. Eukaryotic flagella are less complex than those of prokaryotes. Subcellular components. Illustration depicting major structures inside a eukaryotic animal cell. 17. All cells, whether prokaryotic or eukaryotic, have a membrane that envelops the cell, regulates what moves in and out, selectively permeable, and maintains the electric potential of the cell. Inside the membrane, the cytoplasm takes up most of the cell's volume. All cells, except red blood cells which lack a cell nucleus, and most organelles, to accommodate maximum space for hemoglobin, possess DNA, the hereditary material of genes, and RNA, containing the information necessary to build various proteins such as enzymes, the cell's primary machinery. There are also other kinds of biomolecules in cells. This article lists these primary components of the cell, then briefly describes their function. Membrane. Main article. Cell membrane. The cell membrane, or plasma membrane, is a biological membrane that surrounds the cytoplasm of a cell. In animals, the plasma membrane is the outer boundary of the cell, while in plants and prokaryotes it is usually covered by a cell wall. This membrane serves to separate and protect a cell from its surrounding environment, and is made mostly from a double layer of phospholipids, which are amphiphilic, partly hydrophobic and partly hydrophilic. Hence, the layer is called a phospholipid bilayer, or sometimes a fluid mosaic membrane. Embedded within this membrane is a variety of protein molecules that act as channels and pumps that move different molecules into and out of the cell. 3. The membrane is said to be semi-permeable, in that it can either let a substance, molecule, or ion, pass through freely, pass through to a limited extent, or not pass through at all. Cell surface membranes also contain receptor proteins that allow cells to detect external signaling molecules such as hormones. Cytoskeleton. Main article. Cytoskeleton. A fluorescent image of an endothelial cell. Nuclei are stained blue, mitochondria are stained red, and microfilaments are stained green. The cytoskeleton acts to organize and maintain the cell's shape, anchors organelles in place, helps during endocytosis, the uptake of external materials by a cell, and cytokinesis, the separation of daughter cells after cell division, and moves parts of the cell in processes of growth and mobility. The eukaryotic cytoskeleton is composed of microfilaments, intermediate filaments and microtubules. There are a great number of proteins associated with them, each controlling a cell's structure by directing, bundling, and aligning filaments. 3. The prokaryotic cytoskeleton is less well studied, but is involved in the maintenance of cell shape, polarity, and cytokinesis. 18. The subunit protein of microfilaments is a small monomeric protein called actin. The subunit of microtubules is a dimeric molecule called tubulin. Intermediate filaments are heteropolymers whose subunits vary among the cell types in different tissues. But some of the subunit protein of intermediate filaments include vimentin, desmin, lamin, lamins A, B, and C, keratin, multiple acidic and basic keratins, neurofilament proteins, NFL, NFM. Genetic material. Two different kinds of genetic material exist, deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. Cells use DNA for their long-term information storage. The biological information contained in an organism is encoded in its DNA sequence. 3. RNA is used for information transport, for instance, mRNA, and enzymatic functions, for instance, ribosomal RNA. Transfer RNA, tRNA. Molecules are used to add amino acids during protein translation. Prokaryotic genetic material is organized in a simple circular DNA molecule, the bacterial chromosome, in the nucleoid region of the cytoplasm. Eukaryotic genetic material is divided into different, 3, linear molecules called chromosomes inside a discrete nucleus, usually with additional genetic material in some organelles, like mitochondria and chloroplasts. See endosymbiotic theory. A human cell has genetic material contained in the cell nucleus, the nuclear genome, and in the mitochondria, the mitochondrial genome. In humans, the nuclear genome is divided into 46 linear DNA molecules called chromosomes, including 22 homologous chromosome pairs and a pair of sex chromosomes. The mitochondrial genome is a circular DNA molecule distinct from the nuclear DNA. Although the mitochondrial DNA is very small compared to nuclear chromosomes. 3. It codes for 13 proteins involved in mitochondrial energy production and specific RNAs. Foreign genetic material, most commonly DNA, can also be artificially introduced into the cell by a process called transfection. This can be transient, if the DNA is not inserted into the cell's genome, or stable, if it is. Certain viruses also insert their genetic material into the genome. Organelles. Main article. Organelles are parts of the cell which are adapted and/or specialized for carrying out one or more vital functions, analogous to the organs of the human body, such as the heart, lung, and kidney, with each organ performing a different function. 3. Both eukaryotic and prokaryotic cells have organelles, but prokaryotic organelles are generally simpler and are not membrane bound. There are several types of organelles in a cell. Some, such as the nucleus and Golgi apparatus, are typically solitary, while others, such as mitochondria, chloroplasts, peroxisomes, and lysosomes, can be numerous, hundreds to thousands. The cytosol is the gelatinous fluid that fills the cell and surrounds the organelles. Eukaryotic human cancer cells with nuclei, specifically the DNA, stained blue. The central and rightmost cell are in interface, so the entire nuclei are labeled. The cell on the left is going through mitosis and its DNA has condensed. Cell nucleus, a cell's information center. The cell nucleus is the most conspicuous organelle found in a eukaryotic cell. It houses the cell's chromosomes, and is the place where almost all DNA replication and RNA synthesis, transcription, occur. The nucleus is spherical, and separated from the cytoplasm by a double membrane called the nuclear envelope. The nuclear envelope isolates and protects a cell's DNA from various molecules that could accidentally damage its structure or interfere with its processing. During processing, DNA is transcribed or copied into a special RNA called messenger RNA, mRNA. This mRNA is then transported out of the nucleus, where it is translated into a specific protein molecule. The nucleolus is a specialized region within the nucleus, where ribosome subunits are assembled. In prokaryotes, DNA processing takes place in the cytoplasm. 3. Mitochondria and chloroplasts generate energy for the cell. Mitochondria are self-replicating organelles that occur in various numbers, shapes, and sizes in the cytoplasm of all eukaryotic cells. 3. Respiration occurs in the cell mitochondria, which generate the cell's energy by oxidative phosphorylation, using oxygen to release energy stored in cellular nutrients, typically pertaining to glucose, to generate ATP. Mitochondria multiply by binary fission, like prokaryotes. Chloroplasts can only be found in plants and algae, and they capture the sun's energy to make carbohydrates through photosynthesis. Diagram of an endomembrane system. Endoplasmic reticulum. The endoplasmic reticulum is a transport network for molecules targeted for certain modifications and specific destinations, as compared to molecules that float freely in the cytoplasm. The has two forms. The rough which has ribosomes on its surface that secrete proteins into the and the smoother, which lacks ribosomes. 3. The smoother plays a role in calcium sequestration and release. Golgi apparatus. The primary function of the Golgi apparatus is to process and package the macromolecules such as proteins and lipids that are synthesized by the cell. Lysosomes and peroxisomes. Lysosomes contain digestive enzymes, acid hydrolases. They digest excess or worn-out organelles, food particles, and engulfed viruses or bacteria. Peroxisomes have enzymes that rid the cell of toxic peroxides. The cell could not house these destructive enzymes if they were not contained in a membrane-bound system. 3. Centrosome. The cytoskeleton organizer. The centrosome produces the microtubules of a cell, a key component of the cytoskeleton. It directs the transport through the and the Golgi apparatus. Centrosomes are composed of two centrioles, which separate during cell division and help in the formation of the mitotic spindle. A single centrosome is present in the animal cells. They are also found in some fungi and algae cells. Vacuoles. Vacuoles sequester waste products, and in plant cells store water. They are often described as liquid-filled space, and are surrounded by a membrane. Some cells, most notably amoeba, have contractile vacuoles, which can pump water out of the cell if there is too much water. The vacuoles of plant cells and fungal cells are usually larger than those of animal cells. Eukaryotic and prokaryotic ribosomes. The ribosome is a large complex of RNA and protein molecules. 3. They each consist of two subunits, and act as an assembly line where RNA from the nucleus is used to synthesize proteins from amino acids. Ribosomes can be found either floating freely or bound to a membrane, the rough endoplasmatic reticulum in eukaryotes, or the cell membrane in prokaryotes. 19. Structures outside the cell membrane. Many cells also have structures which exist wholly or partially outside the cell membrane. These structures are notable because they are not protected from the external environment by the semipermeable cell membrane. In order to assemble these structures, their components must be carried across the cell membrane by export processes. Cell wall. Many types of prokaryotic and eukaryotic cells have a cell wall. The cell wall acts to protect the cell mechanically and chemically from its environment, and is an additional layer of protection to the cell membrane. Different types of cell have cell walls made up of different materials. Plant cell walls are primarily made up of cellulose. Fungi cell walls are made up of chitin, and bacteria cell walls are made up of peptidoglycan. Prokaryotic capsule. A gelatinous capsule is present in some bacteria outside the cell membrane and cell wall. The capsule may be polysaccharide as in pneumococci, meningococci, or polypeptide as bacillus anthracis, or hyaluronic acid as in streptococci. Capsules are not marked by normal staining protocols and can be detected by Endurink or methyl blue, which allows for higher contrast between the cells for observation. 20. 87. Flagella. Flagella are organelles for cellular mobility. The bacterial flagellum stretches from cytoplasm through the cell membrane or membranes and extrudes through the cell wall. They are long and thick thread-like appendages, protein in nature. A different type of flagellum is found in archaea and a different type is found in eukaryotes. Fimbria. A fimbria, also known as a pilus, is a short thin hair-like filament found on the surface of bacteria. Fimbriae, or pili, are formed of a protein called pilin, antigenic, and are responsible for attachment of bacteria to specific receptors of human cell adhesion. There are special types of specific pili involved in bacterial conjugation. Cellular processes. Growth and metabolism. Main articles. Cell growth and metabolism. Between successive cell divisions, cells grow through the functioning of cellular metabolism. Cell metabolism is the process by which individual cells process nutrient molecules. Metabolism has two distinct divisions: catabolism, in which the cell breaks down complex molecules to produce energy and reducing power, and anabolism, in which the cell uses energy and reducing power to construct complex molecules and perform other biological functions. Complex sugars consumed by the organism can be broken down into simpler sugar molecules called monosaccharides, such as glucose. Once inside the cell, glucose is broken down to make adenosine triphosphate, ATP, free, a molecule that possesses readily available energy, through two different pathways. Replication. Bacteria divide by binary fission, while eukaryotes divide by mitosis or meiosis. Main article. Cell division. Cell division involves a single cell, called a mother cell, dividing into two daughter cells. This leads to growth in multicellular organisms, the growth of tissue, and to procreation, vegetative reproduction, in unicellular organisms. Prokaryotic cells divide by binary fission, while eukaryotic cells usually undergo a process of nuclear division, called mitosis, followed by division of the cell, called cytokinesis. A diploid cell may also undergo meiosis to produce haploid cells, usually 4. Haploid cells serve as gametes in multicellular organisms, fusing to form new diploid cells. DNA replication, or the process of duplicating a cell's genome, 3, always happens when a cell divides through mitosis or binary fission. This occurs during the S phase of the cell cycle. In meiosis, the DNA is replicated only once, while the cell divides twice. DNA replication only occurs before meiosis. DNA replication does not occur when the cells divide the second time, in meiosis. Second. 21. Replication, like all cellular activities, requires specialized proteins for carrying out the job. 3. Protein synthesis. An overview of protein synthesis. Within the nucleus of the cell, light blue, genes, DNA, dark blue, are transcribed into RNA. This RNA is then subject to post-transcriptional modification and control, resulting in a mature mRNA, red, that is then transported out of the nucleus and into the cytoplasm, peach, where it undergoes translation into a protein. mRNA is translated by ribosomes, purple, that match the 3 base codons of the mRNA to the 3 base anticodons of the appropriate RNA. Newly synthesized proteins, black, are often further modified, such as by binding to an effector molecule, orange, to become fully active. Main article. Protein biosynthesis cells are capable of synthesizing new proteins, which are essential for the modulation and maintenance of cellular activities. This process involves the formation of new protein molecules from amino acid building blocks based on information encoded in DNA/RNA. Protein synthesis generally consists of two major steps: transcription and translation. Transcription is the process where genetic information in DNA is used to produce a complementary RNA strand. This RNA strand is then processed to give messenger RNA, mRNA, which is free to migrate through the cell. mRNA molecules bind to protein RNA complexes called ribosomes located in the cytosol, where they are translated into polypeptide sequences. The ribosome mediates the formation of a polypeptide sequence based on the mRNA sequence. The mRNA sequence directly relates to the polypeptide sequence by binding to transfer RNA, adapter molecules in binding pockets within the ribosome. The new polypeptide then folds into a functional three-dimensional protein molecule. Motility. Main article. Motility. Unicellular organisms can move in order to find food or escape predators. Common mechanisms of motion include flagella and cilia. In multicellular organisms, cells can move during processes such as wound healing, the immune response, and cancer metastasis. For example, in wound healing in animals, white blood cells move to the wound site to kill the microorganisms that cause infection. Cell motility involves many receptors, cross-linking, bundling, binding, adhesion, motor, and other proteins. 22. The process is divided into three steps: protrusion of the leading edge of the cell, adhesion of the leading edge and de-adhesion at the cell body and rear, and cytoskeletal contraction to pull the cell forward. Each step is driven by physical forces generated by unique segments of the cytoskeleton. 23. 24. Multicellularity. Main article. Multicellular organism cell specialization. Staining of a Chenohabditis elegans, which highlights the nuclei of its cells. Multicellular organisms are organisms that consist of more than one cell, in contrast to single-celled organisms. 25. In complex multicellular organisms, cells specialize into different cell types that are adapted to particular functions. In mammals, major cell types include skin cells, muscle cells, neurons, blood cells, fibroblasts, stem cells, and others. Cell types differ both in appearance and function, yet are genetically identical. Cells are able to be of the same genotype, but of different cell type due to the differential expression of the genes they contain. Most distinct cell types arise from a single totipotent cell, called a zygote, that differentiates into hundreds of different cell types during the course of development. Differentiation of cells is driven by different environmental cues, such as cell-cell interaction, and intrinsic differences, such as those caused by the uneven distribution of molecules during division. Origin of multicellularity. Multicellularity has evolved independently at least 25 times. 26, including in some prokaryotes, like cyanobacteria, myxobacteria, actinomycetes, magnetoglobus multicellularis or methanosarcina. However, complex multicellular organisms evolved only in 6 eukaryotic groups: animals, fungi, brown algae, red algae, green algae, and plants. 27. It evolved repeatedly for plants, chloroplastida, once or twice for animals, once for brown algae, and perhaps several times for fungi, slime molds, and red algae. 28. Multicellularity may have evolved from colonies of interdependent organisms, from cellularization, or from organisms in symbiotic relationships. The first evidence of multicellularity is from cyanobacteria-like organisms that lived between 3 and 3.5 billion years ago. 26. Other early fossils of multicellular organisms include the contested Grypania spiralis and the fossils of the black shales of the Paleoproterozoic Francovillian group fossil B formation in Gabon. 29. The evolution of multicellularity from unicellular ancestors has been replicated in the laboratory, in evolution experiments using predation as the selective pressure. 26. Origins. Main article. Evolutionary history of life. The origin of cells has to do with the origin of life, which began the history of life on Earth. Origin of the first cell stromatolites are left behind by cyanobacteria, also called blue-green algae. They are the oldest known fossils of life on Earth. This 1 billion-year-old fossil is from Glacier National Park in the United States. Further information, abiogenesis, and evolution of cells. There are several theories about the origin of small molecules that led to life on the early Earth. They may have been carried to Earth on meteorites, see Murchison meteorite, created at deep-sea vents, or synthesized by lightning in a reducing atmosphere, see Miller-Urey experiment. There is little experimental data defining what the first self-replicating forms were. RNA is thought to be the earliest self-replicating molecule, as it is capable of both storing genetic information and catalyzing chemical reactions, see RNA world hypothesis, but some other entity with the potential to self-replicate could have preceded RNA, such as clay or peptide nucleic acid. 30. Cells emerged at least 3.5 billion years ago. 9, 10, 11. The current belief is that these cells were heterotrophs. The early cell membranes were probably more simple and permeable than modern ones, with only a single fatty acid chain per lipid. Lipids are known to spontaneously form bilayered vesicles in water, and could have preceded RNA, but the first cell membranes could also have been produced by catalytic RNA, or even have required structural proteins, before they could form. 31. Origin of eukaryotic cells. Further information. Evolution of sexual reproduction. The eukaryotic cell seems to have evolved from a symbiotic community of prokaryotic cells. DNA-bearing organelles like the mitochondria and the chloroplasts are descended from ancient symbiotic oxygen-breathing proteobacteria and cyanobacteria, respectively, which were endosymbiosed by an ancestral Archaean prokaryote. There is still considerable debate about whether organelles like the hydrogenosome predated the origin of mitochondria, or vice versa. See the hydrogen hypothesis for the origin of eukaryotic cells. History of Research. Main article, Cell Theory. Hooke's drawing of cells in cork, 1665, 1632-1723. Antony van Leeuwenhoek teaches himself to make lenses, constructs basic optical microscopes and draws protozoa, such as Vorticella from rainwater, and bacteria from his own mouth. 1665. Robert Hooke discovers cells in cork, then in living plant tissue using an early compound microscope. He coins the term cell, from Latin cella, meaning small room. 1. In his book Micrographia, 1665, 32, 1839, Theodor Schwann and Matthias Jacob Schleiden elucidate the principle that plants and animals are made of cells, concluding that cells are a common unit of structure and development, and thus founding the cell theory. 1855. Rudolf Virchow states that new cells come from pre-existing cells by cell division, omnis cellular ex cellular. 1859. The belief that life forms can occur spontaneously, generatio spontanea, is contradicted by Louis Pasteur, 1822-1895, although Francesco Redi had performed an experiment in 1668 that suggested the same conclusion. 1931, Ernst Ruska builds the first transmission electron microscope, TEM, at the University of Berlin. By 1935, he has built an M with twice the resolution of a light microscope, revealing previously unresolvable organelles. 1953, Watson and Crick made their first announcement on the double helix structure of DNA on February 28, 1981. Lynn Margulis published Symbiosis and Cell Evolution, detailing the endosymbiotic theory.